Today we're going to talk about enzymes. Enzymes are a super important part of your body because they are biological catalysts. Why on earth do you need a biological catalyst? Well, let's think about it this way. We turn sugar into energy. It's actually one of the most common cellular respiration equations that you encounter. Sugar C6H12O6 plus O2 is going to convert to water and carbon dioxide. But it's not that easy, is it? I mean, sugar doesn't just sit on my table and react with oxygen in the air and turn into water and carbon dioxide. I can get it to do that in my kitchen, but I've got to crank my stove up to a pretty high temperature. Actually, it's somewhere above 350 Fahrenheit because the difference between sugar caramelizing on your baked good and sugar not caramelizing is the difference between 350 and 375. One of those can melt sugar, one of them cannot. But either way, it's not going to combust it enough to make it CO2. And I wouldn't want to put something that hot inside my body to make it work. So we need fancy enzymatic reactions instead. Enzymes can do the job of catalysts, something as simple as sparking, by manipulating the molecules that they're dealing with such that they're actually easy to break apart. So sugar is a hexagon of carbons. It is not easy to break apart, but a series of enzymes is able to do so. All right, so to understand enzymes, we got to talk about enzymes in a general sense. So that's what we're going to do right now. So enzymes are proteins. Because of that, they are typically big, long, kind of crazy molecules, and they fold onto themselves in a manner that gives them a shape. So we often draw enzymes as some kind of blob, but we'll also give that blob some kind of shape. So this blob has like this round spot in it, but if I drew a very similar blob, only it's got like these different spots in it, it's clearly going to be a different enzyme. Yeah, ignore my dogs barking at the things. So these would both be some kind of enzyme, but they have slightly different shapes. They have slightly different amino acid sequences, all of those things, and they do different jobs. Every enzyme has a substrate. So we've got our enzymes, which are our proteins. We've got our substrate, which is the thing the enzyme can work with. And it's usually shaped pretty similarly to the part of the enzyme that it's going to work with. So, for instance, this enzyme might be connecting to this round thing, and this one might be connecting to two things. One of them a squarish thing, one of them a triangle type thing. Now, we used to think that enzymes had to fit absolutely perfectly, and there was no other possibility for enzymes, but, I mean, biology is squishy, and proteins are squishy. And in fact, most of the time, what happens is the chemicals come close together, and then because chemical bonding always involves some kind of shifting, even if it's hydrogen bonding, which a lot of these reactions are, and it shifts things just so the enzyme kind of moves into place and locks on. This is also what allows for slightly different molecules to occasionally get into the enzyme spot, which is how we can block them, which we'll talk about later. So the key terminology here is that you have an active site in which something goes into, but then also they come together by what we call an induced fit, where the enzyme kind of adapts to pick up the substrate. Once it does that, it's able to react with it. Perhaps it pulls the molecule in such a way so it's easy to break apart. Perhaps it pulls two molecules together in such a way that their electrons tend to connect. Whatever it does, it's able to make that reaction happen, and it's able to do it without there being some kind of complicated, for example, spark going on. So it does it at a lower energy. So lowering the energy or lowering the activation energy of the chemical reaction is really the key part of what the enzyme is doing. In biology, we love to name things in really obvious ways, and enzymes might be the best example of this. So most enzymes are named after their substrate. It's really neat. You might have seen somewhere in a store this stuff called lactase. Sometimes they call it lactade because they want to be fancy, but it's the enzyme lactase. Ace, A-S-E, is the part that means enzyme. So then what's the lact part mean? Huh, sounds like milk, doesn't it? Like lactose. And in fact, lactase breaks down lactose. So lactose is a disaccharide. It is actually a glucose and a galactose molecule stuck together, and your body cannot use them that way, so it needs lactase to break them apart. Many of our digestive systems have lactase, and so it does that job. But if yours doesn't, either you need to take lactase externally in a pill form, or that lactose does not get broken apart and it goes to your colon where the bacteria do nasty things to it. So that's why lactase is important to us. Let's think of some other things you might break apart with an enzyme. For instance, you might break apart a lipid, and therefore I might have a lipase. I might break apart a protein, in which case I might have a protease. Some of the things that it might do too are add or remove certain functional groups. So, for instance, one of the things that is very common is to stick a hydroxyl group or perhaps a carboxyl group onto a molecule. Or sometimes it's even simpler than that, and all we really want to add is a hydrogen. Either way, oh wait, these should seem familiar to you. Like, well, if I add a hydrogen, that's reducing something. If I add oxygens like this, that might be oxidizing something. Hey, I wonder if enzymes are involved in these energetic reactions. Spoiler alert, of course they are. But what would we call them? Well, if it adds a hydrogen, we might call it a hydrogenase. If it adds a hydroxyl, might call it a hydroxylase. And if it adds a carboxyl, might call it a carboxylase. Even if you don't know what the thing is that it's adding, you can figure it out just by the name. Okay, so that's adding. What if I want to remove? Well, if I want to remove, then I'm going to take something off. The slightly different name I might use for this is to take off a hydrogen. I would call it a dehydrogenase. Dehydrogenase. Or perhaps a decarboxylase. This one is actually pretty important. You're going to find a lot of decarboxylases. I would also have a dehydroxylase, but I'm not going to put it here because it's just not going to fit. So those are some enzymes. Sometimes you want to add or remove something a little bit different. So for instance, a phosphatase might add a phosphate group, but also a kinase can add or remove a phosphate group. Well, that's kind of weird. Kinase for phosphate? Well, it's weird, but it still makes biological sense because the KIN there is very similar to kinetic, which is, don't forget, our energy. So like energy of movement, and kinases are very often used when you're dealing with the energy of phosphate groups, for example, ATP and ADP. So keep an eye on kinases for that. Here's another useful one, isomerase. In this case, we're making an isomer of whatever molecule we're dealing with. And here's one more interesting one for you, aromatase. So aromatase makes you think, wait a second, aroma, does it smell like something? Well, kind of, because there's a group of compounds that are known for being a little bit stinky, and in fact, they are benzene rings. So 6 carbons with half of them double bonded, half the bonds double bonded. That's a benzene. Benzenes stink, but they also are called aromatic. See how all the pieces connect? And an aromatase makes aromatic rings. Why would benzene be important in the, biological systems? We don't usually want that. Well, the benzene's not important, but the aromatase is, because there are aromatic compounds in your hormones. And so you want to transfer testosterone to estrogen. Guess what? You need an aromatase. Well, that was an awful lot of different enzymes, but essentially you should be at the point now where you could practically see any enzyme and figure out what it does just by its name, which is a neat trick. A lot of times they also come with the name of the thing they're working with. So for instance, you might get a human pancreatic kinase in some manner, in which case it's from a human and from the pancreas. There's a fairly famous HIV-1 protease, which is obviously from the HIV, human immunodeficiency virus, and cuts apart proteins. And in fact, it cuts the proteins that make the virus. So we've spent a lot of time trying to stop this particular piece. I'll show you that one in a moment. All right. Speaking of stopping things, that is really the next concept, is if we have these enzymes, what if we want to actually make them not work? I mean, you can't have every enzyme in your body working constantly all the time. So let's talk about what happens when you want to make enzymes inhibit or stop their action. Now, sometimes enzymes are active and you want to inhibit them. Sometimes enzymes are actually inhibited and you want to activate them, but they still kind of work the same way. Here are some examples. Human pancreatic lipase. So as a moment ago we talked about this would be in a human. It would come from the pancreas. But this time it's a lipase. So what's it going to do? Break down lipids. So this is a key enzyme in helping you break down the fats in your food. This is approximately what it looks like over here. This is a neat little protein. You can see that it's got these sort of curly parts, the alpha helices. It's got some beta sheets. Don't forget, proteins are complex, but the key concept, of course, is that they have a particular structure. You might notice the one on this side doesn't look quite the same as the one on this side. And that is because it is actually reacting with things. So here you can see this is something trying to react with it, another piece of protein. And up here is what we'd call an inhibitor. It is in the active site, meaning that it is blocking the place where, in this case, the lipid would go. Why would you want to do that? Well, if you were establishing a diet drug, you might want to provide something that blocked lipases, because then the body couldn't break down the fats. Now, you might think that, well, does that help really? The answer is that if you can't break down the fats, you don't absorb them and they don't get into your bloodstream. They simply go out the back end of your digestive system. Now, is it fun to have a lot of lipids go out the back end of your digestive system? Not really. But it does cut back the number of calories that your body's actually using for energy. However, most of these drugs don't really last on the market very well because they make people feel kind of crummy. So you can't really eat all the fat you want and not use it. So here's that HIV protease that I mentioned. Not only is it a very famous protein and enzyme, but it kind of looks like a dog. I love this thing. It's got little mouth and nose and ears, and just in case you don't see it, here it is labeled that way. Now they have it labeled in a slightly different version, the cheek and the whiskers, but I really think it looks like the face of a pug. It's not just because I have a pug, but I mean it's mostly because I have a pug. So what do we do with this? Well, this is a functional HIV protease, and this purple here would be the protein. It comes through, it comes into this spot, and it's supposed to get cleaved, so cut into a way so that the HIV can use it. What do we want to do? Well, we don't want the HIV to be able to use this and make new viruses. We want to block it. So we put in an inhibitor. In both of these cases so far, we've talked mostly about what we would call competitive inhibitors, as in they're competing for the spots on the enzyme. There's only so many enzymes, and there's only so much substrate. And if you can throw enough competitive inhibitor in, you can block up the majority of the enzymes and slow the reaction down. So that is your key. Now, it's not perfect because they are competing, which means sometimes the substrate will get in, which is why, obviously, we haven't solved HIV or AIDS yet, but they do a pretty good job. Protein kinase A. This one is kind of neat because, first of all, it's a kinase. You might recall that's related to the movement of phosphate groups for energy. In this case, is the movement of phosphate groups for energy for proteins, which is like everything in your body. And actually, it's a pretty common molecule and it's pretty important, but it's an easy way to experience activation of enzymes. So this over here is the inactive protein kinase A. So again, it's just a blobby protein type thing. Or up here, you can see the actual blobs of it. Now, the part that's important is the pink part. And what happens to the pink part is that it ends up attached to this other protein. And when it's attached to it, it's stuck. So it's being inhibited mostly what we would call allosterically, meaning that it's connected to it and it changes its shape so it can't work right. When the protein kinase A is attached to the subunit, it doesn't work right, its shape is wrong. But when cyclic AMP comes along, which is something that happens, it's a cell signaler, but also related to the energetics of the cell, it connects to the subunits, which pulls them off. You can see down here, they run away. And then suddenly, the protein kinase changes shape again and is ready to go chop phosphates. What really makes this interesting, of course, is that AMP is adenosine monophosphate, which is a phosphate regulation thing. When there's a lot of monophosphates, in this case, it activates something that might go help you create more ATP, which are triphosphates. So if you have enough energy, nothing happens. But if you're running low on energy, this system allows it to say, oh, we need more. Activate all these enzymes and go make it happen. Here's the aromatase I mentioned earlier. You can see testosterone has a methyl group here and no aromatic ring, but estradiol does. Histone Deacetylase is a good one to tear apart if you haven't tried it before. A histone is a protein in your DNA. Deacetylase. So let's take the ace off. Take the D off because that means we're removing something. And all we've got left is acetyl acetyl, which should make you think maybe acetic acid or something like that. And that's exactly the idea. Acetyl group is exactly the same thing that you would find in acetic acid. And specifically, it is a C with a double bonded O and a CH3. This is the acetyl group. And the histone deacetylase removes it. And then there's an alternate one, the histone acetyltransferase, which actually puts the group back on. So that's kind of neat. You might also notice here that this acetyl is attached to this little extra thing. This extra thing is CoA, that means coenzyme A. A coenzyme is a helper that is needed in this case to make this molecule work. Specifically, it's an organic helper, so it's probably related to a vitamin. The real important part of both of these, though, is that it's connected to your DNA, and if the DNA has the acetyl groups on it, it is allowed to transcribe. If the acetyl groups are taken off, it coils up and says, oh, no, don't. And this is how your DNA decides which things it's going to express and which things it's not. Phenylalanine hydroxylase. So phenylalanine is a thing. Specifically, it's a molecule you could look up. And if you did, what you'd find out is that it's an alanine attached to a phenyl group. Again, all this chemistry, and that's a amino acid. So this is an amino acid, and it's an amino acid that hydroxylase adds a hydroxyl group. Specifically, it turns it into tyrosine, which is an important amino acid. You actually need it for, body functions and stuff. So this is phenylalanine. This is tyrosine. It added this lovely hydroxyl group. Why do I care about this? Well, turns out there are some people who don't have that enzyme. If you don't have phenylalanine hydroxylase, you cannot turn phenylalanine into tyrosine. And it turns out that phenylalanine is a little bit toxic at high levels. And in fact, almost any levels. So if you don't have this particular enzyme, you need to not eat anything with phenylalanine in it. Fortunately, it's not the most common amino acid. It's not one of the 20 everybody knows, and it's only found in very specific things. Next time you are drinking something diet, check out your diet beverage, because a lot of times they say on them, not for people with a particular disease, in this case, phenylketonuria, who are people missing this enzyme. So if you're missing that enzyme, you can't have diet things. So I hope that was interesting and provided you with useful stuff about enzymes.